Hey, listeners of the Bio Report. Before we get started this week, I wanted to tell you about the digital library from Deep Dive. How much time does your team spend looking for research papers? Google, PubMed, social media. There's got to be a better way. You can now search a reference database of 100 million scientific papers and read the full text of 20 million articles, annotate them, and share with colleagues. It's the smarter way to do research. Here's the best part. If you're like me and been frustrated by not being able to access articles you find because they're behind a paywall, I've got good news. With Deep Dive, you get one-stop affordable research. If you're a listener of the Bio Report, you can try the enterprise version of the service for free for 1 month. Go to deepdive.com/podcast and enter the code BioReport. That's Deep Dive, DeepDive.com/podcast. And the code is BioReport, one word, all caps. I'm Daniel Levine, and this is the Bio Report. Osteoarthritis is a progressive disease of the joints for which there is no cure. The condition affects more than 32.5 million adults in the United States, and the incidence is growing as a result of aging, obesity, and sports injuries. Flexion Therapeutics is developing an experimental gene therapy to treat the condition. Rather than correcting an underlying genetic mutation, the experimental therapy, delivered into the joint, codes for the production of the anti-inflammatory protein interleukin-1 receptor antagonist. It is expected to deliver, as needed, anti-inflammatory activity to joint tissues over the long term. We spoke to Mike Clayman, co-founder and CEO of Flexion Therapeutics, about osteoarthritis, the problem with existing therapeutic approaches, and the company's efforts to develop a gene therapy to get the body to produce an anti-inflammatory protein as needed. Mike, thanks for joining us.
A pleasure. Thanks for having me, Danny.
We're going to talk about osteoarthritis, the pain associated with that condition, and Flexion's efforts to address this with a gene therapy in development. Let's start with osteoarthritis. For people not familiar with the condition, what is it?
Osteoarthritis is also known as degenerative joint disease. What is seen is progressive breakdown of the cartilage inside the joint, attendant inflammation, pain, and over time can result in the joint actually becoming no longer as functional as it needs to be, and is often attended by progressive and unrelenting pain. This is a very, very common condition. 30 million Americans have symptomatic osteoarthritis. 15 million Americans have symptomatic osteoarthritis of their knee and see their physicians for that. 5 million patients receive injections into their knee each year for osteoarthritis knee pain, and you cross the threshold for receiving intra-articular injections as a result of exhausting your oral treatment options, which unfortunately also, but understandably, also include opioids. Anywhere from 25 to 40% of patients with symptomatic knee osteoarthritis are treated with opioids. And we identified the osteoarthritis as a very large unmet medical need that was ripe for innovation and which had patients who really wanted and deserved better and longer pain relief than what was currently available on the market. And that's why we've moved in this direction. That's why we developed ZILRETTA. I'll tell you more about that if you'd like. And also why we're developing a gene therapy for osteoarthritis.
You say it's a big unmet need. Can you quantify the market?
Sure. Well, I can quantify the market in terms of the intra-articular, the injection into the knee space market. I said 5 million patients a year receive knee injections. About 4 million or so of those patients receive steroid injections into their knees. None of those steroids are formulated for sustained release, so they flood out of the joint following injection in a matter of hours to days, and the pain relief you get typically wanes after a few weeks. And the other class, hyaluronic acid, it's a naturally occurring lubricant, which provides relatively modest pain relief. And we developed ZILRETTA to address the issue associated with the steroids of inadequate duration of pain relief, particularly since steroids are not injected any more often than every 3 months. 5 million patients receive these injections. If you multiply by the average number of injections per year, it's a total addressable market of 8 million injections a year. And ZILRETTA has been launched, our sustained release steroid that provides, on average, based on our studies, 4 months worth of meaningful pain relief following a single injection. We launched back in 2017, and we're progressively getting ZILRETTA to more and more patients with osteoarthritis knee pain. And we've been very gratified by the results we've seen out there amongst patients who have received ZILRETTA.
How does ZILRETTA differ from existing steroids that are used, and what is do you encapsulate it, or how is it time released?
Yeah.
And how does that change the outcomes for patients?
Sure. So as I mentioned, none of the previously on the market steroids are formulated for sustained release. They're either suspensions or solutions. So there's really nothing that retains the steroid, which is a small molecule within the knee joint. We took a commonly injected steroid, triamcinolone acetonide, married it to a polymer called PLGA, polylactic coglycolic acid, a polymer of two sugars, commonly employed for other products where you want to create a depot, but all of those approved products are injected sub-Q or IM for different conditions using different drugs. What we did was we mixed triamcinolone acetonide with liquid PLGA and then manufactured that as microspheres, which are administered into the joint as a suspension. Once inside the joint, they begin to dissolve, and we engineered these microspheres so that they would dissolve over approximately 3 months. And what we've demonstrated is persistence of low but therapeutic concentration of triamcinolone acetonide in the joint for that 3-month period. And what we were designing for was more durable pain relief. What we observed on osteoarthritis specific measures of pain relief, functional improvement, stiffness improvement, and quality of life was actually better efficacy than was seen with the immediate release steroid. We did head-to-head studies. So we're very pleased with that as a clinical data set. As I say, are moving along in the commercialization of this product to increase the numbers of patients who can benefit from it.
Flexion is developing an experimental gene therapy, FX201. Unlike most gene therapies as people think of them, this is not intended to correct an underlying gene defect, but instead programming the knees to produce an anti-inflammatory as needed. How does FX201 work?
Sure. So, I mean, it's a good intro, Danny, and it's a fundamental difference in the way one thinks about gene therapy. We're delivering a powerful anti-inflammatory protein as a therapeutic. And this is an approach that allows appropriate delivery of that protein for a prolonged period of time based on the preclinical data, at least 6 to 12 months following a single injection. FX201 is a helper-dependent adenovirus serotype 5. The genetic construct within it is IL-1RA, interleukin-1 receptor antagonist, under the control of an inflammation-sensitive promoter. So the idea is the vector is injected into the joint. It's taken up by the cells lining the joint, converting those cells into factories that produce IL-1RA when inflammation is present. Once produced, the IL-1RA suppresses the inflammation and the promoter turns off and the gene stops producing IL-1RA. So that's why we think of it as on-demand anti-inflammatory effect. The helper-dependent nature of this, it means that this vector is non-replicating. We also know that it's non-integrating, and our data to date, preclinically and clinically, would show that it does not distribute outside the joint. By going locally, by injecting this into the joint, a volume of about 5 mLs, one can, at least in concept, dose down aggressively compared to what one would be required if one was trying to achieve systemic concentrations of, say, in this case, IL-1RA, where the systemic volume is 3 orders of magnitude greater than what is seen in the knee joint. And that's important because by being able to dose down, that translates into reduced COGS, which creates less of a burden in terms of the absolute cost of COGS for us, which translates into our ability, we believe, ultimately to deliver a medicine that matters, assuming the data bear this out, to many patients in need.
Has IL-1RA ever been used as a treatment for osteoarthritis previously?
It has been used locally, and there are data that show that IL-1RA injected directly into the joint of patients, directly into the knee joint of patients with knee osteoarthritis, provides a very nice but transient pain relief signal. Because when you inject the naked protein into the joint, there's nothing that keeps it there. So it leaves within a matter of days. And you see waning of the pain relief signal in that over those days. So we looked at that and said to ourselves, if you could deliver IL-1RA persistently and appropriately, you could solve for the issue of transience of presence of IL-1RA in the knee joint. So we were enthused by that. And I think the other thing that more recently has come to light is data from a study called the CANTO study, in which an IL-1 monoclonal antibody, called canakinumab, was being studied in patients who were at high risk for cardiovascular events. And this was many thousands of patients. That study author group looked at a subpopulation of patients who came into the study diagnosed with osteoarthritis. And what they saw was patients who were treated with placebo, and this was as I recall, every 3 months for up to 5 years, either placebo or the monoclonal antibody. They saw a dramatic reduction in joint replacement surgeries for the patients who were treated with IL-1RA, which supports what we've seen preclinically, that not only does it reduce inflammation and relieve symptoms in preclinical models, but it also has evidence of slowing disease progression. So the vision for this product is that we would provide meaningful reductions in pain and improvements in function for at least 6 to 12 months following a single injection, and also have the potential to slow disease progression. And you put all those together, this would be an absolutely transformative medicine in the space.
You're using what's known as a helper-dependent adenovirus as a vector. Certainly familiar with adenovirus vectors, but not with the term helper-dependent. What does that mean, and why the selection of this vector to carry the gene therapy here?
Yeah, we really like the idea of having a non-replicating vector. So you basically remove the genes from the adenovirus that would allow replication, which then begs the question, well, how do you produce it in sufficient quantities if it can't replicate? And that's why you manufacture this helper-dependent adenovirus with a helper virus that supplies the needed protein products to allow replication of this construct. And then you purify out the helper virus, so you're left with just the helper-dependent virus, which can't replicate. And we found that to be quite appealing. And I'll mention that this was not originated by us. This was originated in Dr. Brendan Lee's lab at Baylor College of Medicine. Dr. Lee is a thought leader in gene therapy, and it was his vision initially, originally, to create such a construct. And we chose this because we also saw that in preclinical studies that Dr. Lee's lab conducted, there was better uptake and production of construct in the adenoviral vector than there was in the AAV vector. They did head-to-head studies in joint tissue to look at production of what was enclosed in the vector as a construct. So it was actually chosen quite purposely to be an adenovirus and helper dependent for the reasons I just described.
Is it known yet how localized the effect of the gene therapy is? Does it go elsewhere in the body? And if it does, is there concern about having the protein produced elsewhere?
Yeah, it's a good question. But as I think, just touching on before, by injecting locally and having the vector avidly taken up by the cells lining the joint, at least in concept, there should not be distribution outside the joint. And it turns out that both the preclinical data and the clinical data that we're generating now support that. We do not see distribution outside the joint.
What's the development path forward?
Well, we're in the midst of a single ascending dose study. We're looking at 3 different doses, and we dosed through the low dose, the mid dose, and we're in the midst of dosing the high dose cohort. We've also made the decision to create what we're calling expansion cohorts. The original single ascending dose study had 5 to 8 patients per group. The expansion cohorts will add up to 20 patients per group, so that we can have what I'll call a more robust number of patients to look at in terms of safety, tolerability, and efficacy. What we've guided to is that we will be sharing data from our ongoing efforts by the end of the year. And we just reported on data that we presented at the American Society of Gene and Cell Therapy just a couple of weeks ago that looked at the low dose cohort, evidence of benefit out to 52 weeks in selected patients. I just want to mention this is an open label study, and so it's not as perfectly controlled as one would normally look for, but our view was, in a first-in-man study, let's be sure we're looking at the data in real time, and hence the nature of an open label study. So we're preliminarily encouraged by what we're seeing. We're looking forward to generating more data, and we will look at those data, determine what they're telling us, and make a very thoughtful decision about the continued development of the product, assuming that we see a sufficiently encouraging signal.
And what makes this a compelling indication for this type of a gene therapy?
Well, I mean, if you realize, Danny, once you're diagnosed with knee osteoarthritis, you have a 50% chance of going on to having your knee replaced. And that knee replacement is driven particularly by pain that's grown refractory to available therapies. Not every patient is interested in having surgery. Not every patient is an adequately acceptable candidate in terms of comorbidities for surgery. Anything that could slow or prevent progression to the need for knee replacement would be absolutely transformative in the space. And that's our vision is to get, as I said before, at least 6 to 12 months worth of meaningful pain relief and functional improvement from this product. There is nothing out there that would that is similar at all to this, and it would represent best in class should it fulfill that. Even that, and it would be one step above even that vision if we had evidence of slowing of disease progression. It's a tremendous unmet medical need. There are over 1 million total knee replacements each year, and people have them out of the fact that their lives have become compromised for reasons of pain and functional compromise.
Like other gene therapies, this is expected to be a one-and-done treatment. I expect it'll carry a relatively high price tag compared to existing therapies, but beyond the clinical data, are you thinking about other data you'll need to collect to make a case to payers?
Yeah, I can tell you that health econ and outcomes research perspective is being applied as we speak, and we're being very thoughtful about constructing value propositions that will make sense. I think it's premature for me to go into dollars and cents, but I'm confident that if this product delivers as we hope it will, we will find a path forward that will be embraced by patients, physicians, and payers.
Michael Clayman, co-founder and CEO of Flexion Therapeutics. Mike, thanks so much for your time today.
Danny, a real pleasure. Thank you.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.